So thinking about ER protein import can get a little complicated because there are certain parameters that have to be observed when considering where a protein ends up, whether it's a transmembrane protein or a soluble protein, whether or not it's a single pass or multi-pass, and what the orientation is of those proteins. And so what I've done is I've developed a little demonstration using a rope and some pool noodles and this little wooden contraption here. And as the next few moments of the video will explain, this is the ER membrane. We're going to use this here to demonstrate or to represent proteins with various sorting signals on them. So watch the next few minutes, get a good understanding of what the setup is here with this little piece of wood, as well as what the different pool noodles are and what the colors mean and that sort of thing. And then we'll go through a couple of examples and just kind of give you some sort of visualization of what's going on with protein import. So I'll see you then. Okay, so the piece of wood that you saw is going to represent the ER membrane. And for these demonstrations, we're going to identify the space above the ER membrane as the cytosol. The space below the ER membrane is going to be designated the ER lumen, or the luminal space of the ER. You'll also notice a little gray patch on the left side of the ER membrane. This is meant to represent the translocator. So anytime we insert anything into the ER membrane, it's going to be at that area where the translocator is. So hopefully this gives you some orientation so that as you watch these demonstrations, it will clarify what's going on at the ER membrane in terms of ER transport. If you have any questions, let me know. Thanks. So before each demonstration, I'm going to show you an image that is meant to represent the signal sequence arrangement on a particular protein, on the protein that we're going to be discussing in that particular segment of the video. And so all the images will start out looking essentially like a straight line, which is meant to represent the primary structure of the protein for that particular demonstration. The N and C termini of the protein will be indicated by an N and the letter C on the left and right side of the line respectively. And then you'll see a series of colored boxes. Now these boxes also correspond with the color of the pool noodles that I'm using on the rope in each demonstration, so you can have an image to kind of remind yourself of which colors mean or represent which signal sequences. And so what I want to do is just go through and just kind of give you an introduction into what those sequences are. So the first one, the red pool noodle, will indicate a start, and start in this case represents the ER start transfer sequence. And remember, this is the same thing as an ER signal sequence. The orange pool noodle will represent a stop, or an ER stop transfer sequence. Purple pool noodles represent a nuclear localization or a nuclear import sequence, same thing. The yellow pool noodle will represent the ER retention sequence. Green pool noodles will represent mitochondrial localization sequences. And then lastly, the blue will represent a mannose-6-phosphate sequence, which, as you hopefully recall, is a lysosomal targeting sequence. And so in each demonstration, if you forget which sequence is what, you can just bring up this image, and I'll make sure that this image is available
for your reference for each of these demonstrations. The first example that we'll look at is a protein with just a simple ER start signal sequence at the N-terminus. This is the same protein that can be found in figure 1515 of your book, and it's also one that I covered in the lecture over chapter 15, that very first example.
Okay, so we're starting out with a protein that has a single ER start signal at the N-terminus. So I'm going to pretend I'm a ribosome and I'm away from the ER and I'm starting to translate a protein. And so out pops the start. So again, I'm a free ribosome. Out pops the start. Immediately what happens? You should be screaming the SRP binds, right? So the SRP, the signal recognition particle, binds that start sequence, locks up the ribosome and prevents it from, halts translation. We go over to the ER membrane. You got to remember where my translocator is, and the SRP is going to bind to the SRP receptor, and it's going to insert the start signal in such a way that the N-terminus, which is the first part that pops out of the ribosome, so this is the N-terminus, so the N-terminus is facing the cytosol, and remember this space is the cytosol. So it's going to insert it just like this. Okay? Then what happens? Translation continues, or it restarts, and as I'm translating, the protein is being translocated into the ribosome, into the ER in the lumen side, and here is the C-terminus. And we have one more thing that we have to consider, because remember, this is a start sequence and it's at the N-terminal. It's not internal, but N-terminal. So what happens? Everybody should be screaming it gets cut off, right? So I'm going to pretend that this gets cut off. And now, hopefully you can see, we've got our soluble protein on the luminal side. This gets kicked out and it goes away, it gets degraded. So we have our soluble membrane protein down here in the ER lumen.
The next example that we're going to cover is a protein with an N-terminal ER start signal sequence, followed by an ER stop transfer sequence. This is the same protein that is diagrammed in figure 1516 in your book, and is also the same one that I cover as the second example in the same lecture over chapter 15.
Okay, for this next example, we've got an N-terminal start sequence and an internal stop. And these are both, it's an ER start signal sequence and then a stop transfer sequence. So again, I'm going to be a ribosome. Everything always starts out in the cytoplasm with a free ribosome. And I'm translating, and then out pops the start. The SRP is going to bind. It's going to take us over to the ER. This is the N-terminal, so remember that's always the side of the protein, or the end of the protein that pops out of the ribosome first. So we said this was an N-terminal start sequence. So it's going to insert like this. Okay, remember the N-terminal side is going to face the cytosol. Translating and translocating at the same time. Out slides the stop, And this is what I want you to see. So this is gonna slide right in just like that, okay? I'm gonna be translating, and I'm translating, still translating. Out pops the C-terminal end, okay? So the C-terminal end, at least right now, is in the cytoplasm. We have one more thing left to do, right? And that should be to cleave off the start signal sequence, because it's an N-terminal start sequence. So we're going to cleave that off. That gets degraded, goes away. And now we have a protein, a single pass transmembrane protein with the N-terminal side. I don't think you can see it, but the N-terminal side on the lumen and the C-terminal side on in the cytoplasm. Okay, and then this would get kicked out from the translocator, just like this, and there's our final protein. Now, if this happened to go to the cell membrane, which right now, there's nothing keeping it in the ER membrane. So remember, the default location for any of these, if there's not that second signal, is the cell membrane. And so if that were the case, then this protein would be a transmembrane, single pass transmembrane protein with the N-terminus outside the cell and the C-terminus inside the cell on the cytosolic side. Because remember, whatever's out here on the cytosolic side stays on the cytosolic side, whether it's in the ER membrane, the Golgi, in a vesicle, in the cell membrane. So if we had a cell membrane up here, this would be hanging out in the cytosol. So we'd essentially just take this, turn it upside down, and that's what it would look like in the cell membrane. Okay?
The next example that we're going to look at is a protein with an internal ER start transfer sequence, or a start, followed by an ER stop transfer signal sequence. This is the same protein that's diagrammed in figure 1517 in your book, and it's also the third example that I cover in the lecture on chapter 15 as well.
So this next one, we have an internal start, ER start signal sequence, and we have a stop that follows that. So again, I'm a free ribosome out in the cytosol. I hop onto my RNA, mRNA specifically, start to translate. Nothing's going on, nothing's happening, still translating, and then out pops the start. And what happens? The SRP is going to bind to the start. It's going to stop translation. Again, not going to terminate it, but it's going to stop it or halt it for a moment. Carry that whole complex over to the ER, and again, we're going to insert the start sequence so that the N-terminal side, which is this side, okay, because that's the side that's closest to the N-terminus, we're going to insert it so that the N-terminus is facing the cytosol, just like this. SRP dissociates, translation starts back up, translating and translocating all at the same time. Out pops the stop signal sequence, or the stop sequence. It's going to slide right into the translocator just like that. I'm going to finish translating, and then this is what I've got. Okay, this gets kicked out. I'm going to try to move this without it breaking down. This gets kicked out just like that, out of the translocator. Now, is there anything else that we need to do? Hopefully you said no. If you said yes, that we need to cut off the start, ER start signal sequence, remember, this is an internal signal sequence. We don't cut those off. We only cut them off if they're at the N-terminus, like the first 8 to 10 amino acids. This is an internal signal sequence. So nothing gets cut off. And so now we've got a multipass transmembrane protein that crosses the membrane twice. We have both the N terminus and the C terminus in the cytosol with the loop in the lumen. There's nothing else holding it into, or there's nothing else holding it in the ER membrane. There's no other signals. So this would go to the cell membrane, and we would have the loop on the outside of the cell. And again, if it's on the cytosol, when it gets inserted in, or in the cytosol when it gets inserted into the ER membrane, then it stays in the cytosol wherever it ends up. And so the N terminus and the C terminus are going to be hanging down, let's say the cell membrane is up here. The C terminus and the N terminus are going to be hanging down in the cytosol. All right?
In example 4, we've got a protein with an N-terminal ER start signal sequence followed by a mannose 6-phosphate site. So this is going to be a little interesting. So what I want you to do is pay attention to not only how it inserts into the ER, but also think about where this is going to end up at its final destination. And that's going to be covered in the next segment of this video.
Okay, so the next example we have is a protein that has an N-terminal ER start transfer sequence, or START, and an M6P, a mannose 6-phosphate, which is going to tell the cell this protein needs to go to the lysosome. So again, we're always going to start at a ribosome in the cytoplasm. That's free in the cytoplasm. I'm going to start translating. Out pops the START. That's going to cause the SRP to bind. We're going to come over to the ER. This is the N-terminal side. Technical difficulty. And this is going to insert so that the N-terminus is facing the cytosol. So it's going to insert like this. Okay. Translation begins, or starts up again. Out pops the mannose 6-phosphate. Nothing happens. This is going to pass through. Now this is not a hydrophobic sequence, and I think that's what sometimes will confuse people, is the only hydrophobic sequences that we ever talk about are the ER starts and the stops. Okay, so this is going to pass through just like this. The rest of the protein is going to go through. Here's our C-terminal end, and we should be screaming at this point that the ER start sequence gets cut off. So we're going to cut that off. This is going to get degraded. Now we have a soluble protein here with a mannose 6-phosphate signal sequence on it. So this is going to definitely leave the ER, and this is going to go to the lysosome, and this will be a lysosome lumen protein, not a membrane protein, but a protein that lives and works in the lumen of the lysosome. And the reason why is because we have this second signal that tells it where to go once it's in the endomembrane system.
In this example, we've got quite a mix of different sorting signals. We got a nuclear localization signal sequence, or a nuclear import sequence, followed by an ER start signal sequence, and this would be an internal start sequence. And at the very end, we've got a KDEL signal sequence, which is an ER retention sequence. So watch the next segment of this video to see what eventually happens to this protein.
Okay, so we've got a protein that's going to have a nuclear localization sequence, an ER start, and a KDEL, which remember a KDEL is an ER retention signal. So again, I'm a free ribosome out in the cytosol. I just hopped onto an mRNA, and I'm starting to translate, and out pops an NLS, nuclear localization signal, but nothing happens, right? Because the only time the SRP is going to bind is if it's a start signal. So I'm still out in the ribosome. I'm a ribosome still out in the cytoplasm, and I'm just translating away. And then out pops this start signal. Now the SRP binds. Okay? So the SRP binds. I'm going to come over to my ER membrane in the translocator. This is the N-terminal side of the start sequence, and so that's going to face the cytosol. So it's going to insert like this. Now this is still out here. So translation is going to continue. Translocation is going to start back up as well. Here comes my KDEL. Again, not a hydrophobic sequence. Only the start and the stops are hydrophobic sequences. So that's going to just pass right on through. And that's going to end up in the lumen. Okay? Now we have to ask ourselves a couple of questions. What does this do? Well, remember the NLS is only going to send proteins to the nucleus. But remember, nuclear transport is post-translational. So since there was a start, what happened is the protein finished, or the ribosome finished translating the protein in the membrane of the ER. And remember, once a protein is in the endomembrane system, whether it's the ER, Golgi, or whatever, it's not going to be able to leave. So in order for this protein to get to the nucleus, it has to somehow detach or come out of the ER membrane or leave the endomembrane system to go to the nucleus, and that just doesn't happen. So what we could do with this is we could just ignore it. Okay, now it doesn't get cut off. I want to make sure we understand that's not what I'm doing here. I'm just taking this away because I can just ignore it. Okay, it's still there, but it has no bearing any longer on where this protein is going to go because I'm already locked into the endomembrane system. Okay. Now, what I need to ask myself is, okay, well, what about this start sequence here? Well, it's an internal start sequence, so it does not get cut off. So it's just going to get kicked out, just like this. Okay? So now we know that we have a single pass transmembrane protein. The N-terminus is in the cytoplasm. The C-terminus is in the lumen. And we also have a KDEL. Okay, so remember the yellow pool noodle represented a KDEL sequence. And the KDEL sequence is an ER retention signal sequence. So what this means is that the final destination for this protein is, in fact, the ER membrane. Now, yes, it may get caught up in a vesicle formation and be sent to the Golgi, but remember this KDEL sequence is going to get it back to the ER. So we would call this an ER resident protein that is a single pass transmembrane protein with the N-terminus in the cytosol and the C-terminus floating around in the lumen of the ER.
In this last example, we take the same protein from example 5 and switch out that internal ER start sequence with a mannose 6-phosphate. So what we're going to start with or what we have is a protein that has a nuclear localization signal or nuclear import sequence, followed by a mannose-6-phosphate site, ending with a KDEL sequence. So this should be very interesting, and I'd encourage you to think about ahead of time where this might end up, what it might look like, before you watch the rest of the video and see if your prediction matches up.
Okay, for this last example, we have a protein with an NLS, a mannose-6-phosphate signal sequence, which is a lysosomal targeting sequence, and a KDEL. So again, as always, I'm going to start out as a free ribosome in the cytosol. I'm going to start translating. Out pops my nuclear localization sequence. My question to you is, does anything happen? The answer should be no, because it's not a start sequence. So the only time something happens that would cause any sort of change or diversion from what's going on with regular translation is if a start sequence pops out. So I'm going to start, just continue, okay? And then out pops my mannose-6-phosphate, okay? Does anything happen at this point? No, not at all. Now you might be saying, but it's a mannose-6-phosphate site. Shouldn't it go to the ER? No, it does not go to the ER unless a start pops out, okay? And that's not what's happened. The nuclear localization sequence that I mentioned earlier, you may have asked yourself, and I neglected to mention this earlier, but would this cause it to go to the nucleus? Not at this point, okay? Because we're not finished translating. And remember, nuclear transport and mitochondrial transport are post-translational. So the protein has to finish translating before it can be transported. And we're not finished translating. So right now, we're not worried about this, okay? And we're not worried about this either just yet. Now, I'm continuing to translate, and out pops our KDEL sequence. Again, is this a start? Are we supposed to go to the ER membrane at this point? And the answer should be no, because it's not a start. Well, now I'm finished. So now I've got a protein that has a nuclear localization sequence, a mannose-6-phosphate, and a KDEL. So now what I do is I now reevaluate my signal sequences. And I'm just going to start at the beginning. Does the nuclear localization sequence have any bearing on where this goes at this point? We're finished translating. And the answer is yes. This could tell the cell, hey, this needs to go to the nucleus, okay? What about this mannose-6-phosphate signal? Does this have any bearing? It doesn't, because the only way this would be of any concern is if we were in the endomembrane system already. And we're not. We never had a start. So I can just ignore this. Again, I'm not cutting it out. It's still there. I'm just removing the noodle to let us know that we can ignore that. Okay? Same thing with the KDEL. Does this have any bearing on where the protein goes? Well, it doesn't, because this is an ER retention signal sequence. It has to be in the ER, or at least in the endomembrane system, in order for this to have any effect on where it goes. So we can ignore this as well. Again, it's not getting cut off, it's still there. We're just removing it to let us know. We don't have to consider that. So now let's go back and look at what we have left. Well, we've got a protein that simply has a nuclear localization signal. And that's the only, well, it still has the other two, but this is the only one we need to really be concerned about. So this is going to go to the nucleus. Now, remember, when it comes to the nucleus and the mitochondria, we don't get up in arms about is it in the membrane, is it in the lumen, or anything like that. We would just say that this goes to the nucleus, okay? So again, the key things that we need to understand about when we're solving these types of problems is we've got to first remember everything, all translation starts on a ribosome that is free in the cytoplasm, okay? It only goes to the ER if a start signal sequence pops out. either an N-terminal or an internal. Once translation is finished, then you reevaluate your signals, okay? So if you are a protein and you're in the ER immediately after translation, anything that says go to the nucleus or the mitochondria, you can ignore because you can't get back out in order to go to those organelles. Anything that tells you to stay in the ER, go to the lysosome, or if there's nothing there, you can reevaluate. Okay, so if there's a KDEL, it stays in the ER. If there's an M6P, then it goes to the lysosome. If there's nothing else except a start and a stop or some combination of starts and stops, then it goes to the cell membrane. So remember, the default location is not the ER. The default location is the cell membrane. Now, if I never have a start, I would do the same thing. I would finish translation out in the cytoplasm, and then I do just like we did here. I go back and I reevaluate my signals. Does the nuclear localization signal still have an impact on where it goes? Absolutely. If I had a mitochondrial signal sequence, would that have any bearing? Absolutely. Now, I'm not going to give you, now you may be asking yourself, well, what if it has a nuclear localization signal sequence and a mitochondrial signal sequence? After translation, I wouldn't do that to you. I'm not even sure where it would go if one would win out over the other. But if I have anything that tells it to stay in the ER or to go to the lysosome, I can ignore those because I'm not even in the endomembrane system.
Hey guys, thanks for watching the video. I hope you learned a little bit more about how proteins get to the various organelles of the cell. Those examples that we just looked at are not the only ones possible. In fact, I encourage you to maybe come up with your own. Maybe pick some at random, or think about where you might want to have a protein go and what are the right mix of signals that you would need to get that protein to go to that specific location. In the end, though, I just want you to think about the whole process of how these proteins get sorted to the different organelles. And the main thing that we need to always remember is that all protein import, regardless of where it goes, whether it goes to the nucleus, it goes to the lysosome, if it gets secreted, or if it stays in the cytoplasm, all begins on a ribosome that's free in the cytoplasm. So think about the process as you maybe interrogate different mixes of signals. And at the end of the day, if you have any questions, you can certainly email me at rabennett@usi.edu. And again, I hope you enjoyed the video. It was fun making it.